research method and design. So there are two broad approaches to research and science. One is the traditional research process, which you may be familiar with when thinking about the basic scientific method. And it generally begins with a research question or hypothesis. And sometimes people will talk about, first there is kind of an observation or a hunch, right? And then that leads to a research question or hypothesis. And then from that, you design a study and collect data to address that hypothesis. And then after you collect the data, you summarize the data with some summary statistics and visualize the data. And then you use more sophisticated methods to draw inferences from the data, such as whether or not a variable is causing another variable. And then you write up the results in a paper and you communicate the results of how you answered that hypothesis. Now, another approach that is more common nowadays and kind of gets mixed up with this traditional approach is the computational approach of research design. And I call it computational because it is based on the availability of more data nowadays than before, and also means and the tools with which to process the data much more quickly, and therefore the ability to test all sorts of associations on the data. Now, this creates actually kind of issues about the nature of inference and the nature of science itself that we'll probably not get into this course, but you may want to think about. But what happens nowadays is we'll have data collected already, maybe for a specific study, or maybe just general data collected for general purposes. And we'll import the data into a program like R, and then we'll import it into a kind of another more specialized program like Tidy, which can organize and do analysis and visualizations on the data. And then we'll actually iterate in this kind of exploration quadrant here, where we transform the data, we visualize the data in different ways, and you model the data in different ways. So maybe you test whether or not one variable is affecting another variable. And if that doesn't work, then you add more variables, or you remove some things, you add other things. Some people have noted, once again, problems with this approach, but it's very common nowadays. And it also has its benefits because it enables us to see the patterns on the data and visualize different configurations of the data, and then actually address research questions or relevant hypotheses that we may have. When we talk about statistical analysis, there are just two broad approaches, and you can think of this as two broad approaches of statistical analysis, or two broad types of statistical approaches. So that's the exploratory and the inferential. The exploratory is also called descriptive, and that's the methods for summarizing the existing attributes of the sample of the data, or maybe of the population of the data as well. So this is the calculation of what we also call summary statistics, and includes getting measures such as average of an attribute or percentage of a category. So for example, let's say you're collecting data, you're surveying people about, and you want to know their races. That's part of the survey. And you identify the percent of people surveyed that identify as white, percent of people surveyed that identified as Black or Hispanic, and so on. So those are what we call the descriptive or summary statistics. Average age of the population survey, the average shares of each post. These are kind of the basic exploratory or descriptive measurements. They are statistical calculations. The average, the mean average, is a formula, is a calculation, but it doesn't, by itself, it does not enable us to make inferences, farther inferences about the information we're observing. And the inferential statistics are the set of methods for drawing conclusions of two kinds. We're really only doing two things when we're making inferences. One is generalizability, So that is, we're trying to investigate the extent to which the sample can be said to reflect the population. So because we often are dealing with sample and not the whole population, we want to make sure that the sample that we have, the group that we of individuals that we were able to acquire, can actually represent the population. That's what we mean by generalizability. The sample has to be generalizable to the population or representative of the population. And the other, I guess, more difficult inference is that of, but also more important and more interesting and more valuable, is that of causality, which is the extent to which we can say with confidence that one variable is causing another. It's literally directly or indirectly impacting another variable. And we will explore this. This is what we will explore throughout the whole course. So we're just kind of taking a broad view here, and then let's talk about the two main types of studies also in very general, how we approach a scientific or a research study. And there's two main ones. One is what we call the observational study, and that is when data is collected without a direct intervention or interference in the phenomenon being observed or in the people that you're collecting from or in the things that you're observing. So things like interviews, surveys, collection of blood samples, counting of events. You are collecting the information from them. You might be asking a person to fill out a survey. You might be actually drawing blood from a person, but this is not an intervention. An intervention is when you actually modify some condition for a group, generally a group of people, to observe differences where the intervention was included from where the intervention was not included. And that is what we call an experimental study. The experimental studies is when there is a direct intervention or treatment and a separation between the two groups, and you observe differences between where the treatment was placed and where the treatment was not placed. So the treatment group receives the intervention and the control group does not receive the intervention. And this is often discussed in terms of medical work, but it doesn't have to be. It can be in other instances as well. We can be for psychological studies. We do this for even sometimes sociological studies, for studies of information systems. When we talk about A/B testing, one group of people use the new system, and one group of people, which is the new system in this case is the treatment, is the new intervention, and then a group of people use the old traditional system, and that's the control group. So those are the observational study and experimental study. Also, we can break down further observational studies. So from observational studies, there are prospective studies, and these collect information as events unfold. And prospective meaning that we actually already developed the study, and then we go collect the data. So medical researchers may identify and follow a group of patients over years to assess the influence of behavior on cancer risk. So we're kind of like developing the study ahead of time and then go collect the data. Now there's retrospective study, which is essentially you're using data that was already collected. So there's some data over there. There's data about maybe our patients and their conditions and if they had cancer or not, or maybe they have diabetes or not. And then from the existing data, we recognize that we might be able to do a study and check for associations or causality of some variable on another. And in experimental studies, well, it gets more detailed as well when we're talking about how do you actually configure and set up an experimental study. But generally, there are two groups. One receives a treatment, what we call the intervention, and one receives a placebo. Now, some studies don't actually give a placebo, but we really should because it helps the study. And the placebo is the thing that the treatment that one of the group, the control group, thinks it's receiving, because if they don't receive something, then it's kind of difficult to compare with the other group that did receive something and are aware that they are using, for example, the new information system rather than the old one. So multiple groups also may be divided into these two, control and treatment. So often we may not just have a treatment and a control group, but if there's another variable that is very influential in impacting your outcome of interest, then we may actually have to separate the population in two groups. So for example, when we're talking about COVID-19 or maybe cancer, we can't just take the whole population and test maybe a new treatment on the whole population as a whole with treatment and control. We may need to create blocks of a low-risk population and a high-risk population. And within these two blocks, we have a control group and a treatment group, so we can better assess the effect of the treatment in the different populations. In the low risk, maybe it has one effect. In the high risk, it has a different effect. So if we put them together, we may blur out and not be able to see this relationship. Ultimately, I want to end with this scope of inference chart here, which I find useful from our textbook. And it shows how we can really make this inference, draw this kind of inference of generalizability and causality from our study and our research. And random sample is really important, and it helps us be able to determine causal relationship and generalize our sample information to a population. And whether or not we have an experimental study or an observational study too is going to impact whether or not we can generalize and identify causal relationships, or we cannot do any of these things. So I want you to explore and see further details in the Montana State textbooks that we're using for this course. And I'll see you next time.